drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content.
Thanks for having me.
Can you start off by just telling everyone a little bit about who you are and what you study?
Sure. Well, I'm an associate professor of medicine at Baylor College of Medicine. And since the earliest days of my career, I've been studying kind of repair and regeneration. And that led to kind of just when I say repair and regeneration, I mean everything from limb deformities in amphibians to pheromonal communication in terrestrial newts to bioengineering tissues. And then my professional career has really focused on injury repair and the injury repair process in particular in the context of aging. So why don't we repair as well when we age? And why do we have age-related diseases, or why do we have certain diseases that are much more prevalent in the elderly and or worse outcomes with certain diseases in the elderly, and how can we target those processes so that the elderly can repair or can repair or have better outcomes more similar to a young person.
Right. Yeah, I actually just had Michael Levin on, and a lot of our discussion centered on regeneration. And basically, one of the things that's really interesting about this field, when we're thinking about vertebrates at least, is we all have regenerative capabilities, but those basically go down as we age. So, he made the point that you can split a human embryo in half when it's at the one cell stage and it becomes two full humans. But then as we age, right, that regenerative capacity goes down. And then everybody knows at an intuitive level that at some point you get old enough where you struggle to even repair simple damage to the body. And so something age-related has happened there where the body's reparative mechanisms go down just steadily over time, right?
Exactly. Yeah.
So,
a good example of that is, I study fibrosis and fibroblasts. Fibroblasts are like the key cell that generates scar tissue. And so scar tissue tends to be, diseases of scar tissue or fibrotic diseases tend to be more prevalent as we get older. And so one of the things that we've found is that that key cell, the fibroblast, has this enormous amount of plasticity. It can differentiate into a myofibroblast and generate scar, and then it can de-differentiate back into its precursor. And that process tends to be impaired as we age and in the context of disease as well.
See, so these fibroblasts are a common cell type, and they're the thing that makes scar tissue, basically.
Exactly.
So they can differentiate into cells of the body. Like if you've got a scar on your skin, they can turn into new skin cells that are composing the scar tissue, and then they can also de-differentiate. So they're kind of like stem cells or something.
Yeah, they're this kind of master reparative cell. They're sort of dormant in your tissues until you need them. Mm-hmm. And so like when you fall and scrape your knee, then your first line defense is your immune system, inflammation happens. And then your second line defense is that these fibroblasts proliferate, they migrate toward the site of damage. And then they differentiate into, fibroblasts differentiate into myofibroblasts, which are muscle-like cells. And then why do you need muscle-like cells? You need something to pull that wound closed. And cells generate that scar tissue matrix. And then as the wound is resolving, those cells either undergo apoptosis or death, or they de-differentiate back into their precursor. And so that process of of either they become resistant to apoptosis or they fail to dedifferentiate back. That is one of the things that happens that contributes to this persistent scar tissue response.
I see. So these things are dormant. Where do they come from? Are they sort of locally embedded everywhere? So if I get a
Yeah,
cut on my,
fibroblasts are in almost every tissue, and
okay, okay,
they're
so they're sort of ready and waiting everywhere.
ready and waiting. Mhm.
So you get a scar. So first you get some kind of injury, and you literally have to plug the hole and like stop the initial problem. And so they are literally turning into something like a muscle cell, and then pulling the wound closed, essentially.
Yes. Yep. Yep. While
Interesting.
they're generating scar, and then as that process, as you're resolving that injury, those cells will again either die, which is a
Hmm.
normal part of the repair process, or they'll dedifferentiate back into that precursor cell.
Yeah.
So that's actually one of our findings that we made was that those cells do have the capacity to dedifferentiate, and that on top of that, that capacity can be impaired when we age.
Hmm. So if I get a cut on my skin or something, the wound will essentially be pulled closed because these fibroblasts will turn into muscle-like cells to hold it closed. My new skin cells will form and everything will go back to the way it was, almost, depending on how old I am and how well the process is working. And then those cells will either, the fibroblasts that helped pull the wound closed, they will either die off or turn back into fibroblasts because they're no longer needed there.
Right.
So what starts to happen when that doesn't happen? We get older and they don't dedifferentiate or
Yeah.
apoptose away.
Yeah. Okay. So I study this exactly, fibrotic diseases. Um, and so another example of that is liver cirrhosis. A lot of people have heard of liver cirrhosis. End stage heart failure is often caused by progressive scar tissue.
Mm-hmm.
And so the same thing is happening. Like when you lose blood flow to your heart, we can easily medically open up that blood flow again with a stent, get blood flow back to your heart. But where you have that scar, that myocardial infarction, that scarred area, the problem there is that it continues to grow, and so your heart is continuing to remodel. So as you get more scar tissue, contractile cells over here, your heart's expanding over here, and so your heart is always, your heart is remodeling
Yeah.
as this process progresses until you ultimately have end stage heart failure. I
So,
study a disease, a rare disease where it's the most fatal and progressive disease of scar tissue, which happens in the lungs.
yeah. So basically, when you're relatively young, it sounds like you can get scarring in any tissue. When everything is working, as it should, you will get very close to recapitulating the anatomic and just biophysical and spatial. You'll recapitulate the tissue as it was, and you'll have very little physical difference there. But as we age, the repair process starts to break down itself, and then you have scar tissue, say in your heart or your liver, wherever, and it might be kind of close to the way the tissue was before, but not quite. And then maybe you start layering on more scar tissue on top of it, and that's just sort of physically, anatomically, just changing things, pushing against cells, not allowing the blood to flow through the tube
Right.
quite as much, like all of that stuff.
Yeah, exactly. And keep in mind too, like each organ has a different capacity for turnover and repair. Like obviously
Right.
skin has a better capacity to turn this over than heart, right?
Mm-hmm.
So inherently different tissues have different capacity for turnover and repair. But in general, that process tends to be less and less and less efficient as we age.
Yeah. So is it possible to, like, if you get a cut on your skin, say, and then you just observe how prominent the scar is after it's healed, does that tell you something about how far along in this aging process you are?
It's a good question. I mean, I don't know the answer exactly, but I would imagine that, like, and I'm sure you can imagine too, like if you're 10 and you fall and scrape your knee and you're 50 and fall, I mean,
Right.
I would imagine that as we age, that scar is going to be more prominent. I don't know the answer though, but I would assume that's going to be the case.
Yeah. Yeah. And so let's talk about the recent paper you did, and maybe we can circle back to some other stuff that's to do with fibrosis and oxidative stress more generally. But you just did a study that people have been talking about, because you did something looking at psilocybin, which is the precursor to psilocin. So this is the active compound in magic mushrooms. You eat mushrooms or something with psilocybin, your body turns it into psilocin. It's 5-HT2A receptor agonist. I've talked about this many times on the podcast. It's the thing in magic mushrooms. But you looked at it in a way that I don't think anyone else has looked at it, or it's not known for, which is in the context of longevity. So let's just start out really basic here. What was the study in terms of why you even thought to do this and what the setup is?
Sure, yeah. Well, you know, I've obviously heard of psilocybin. Most people have heard of psilocybin. It's out there. People are talking about it. There are lots of podcasts, Netflix shows talking about psilocybin. And this is really how it started. Just a friend of mine who's not a scientist, just kept asking me questions about psilocybin. And, this when you're a scientist, I think your non-scientist friends kind of always view you as like the all science knowing person. They don't realize that we don't really know much at all. We just know a lot about one little thing or the area that we study. So he was asking me all these scientific questions about psilocybin that I didn't know the answer to. And I just decided for him that I would just do some reading. And when I say reading, I mean I only wanted to look at peer-reviewed publications, like what is actually scientifically accepted and known about psilocybin.
I'm familiar with the experience.
Yeah, and so I realized there were not that many studies actually on psilocybin. I mean, relatively speaking, like if you were to compare it with like resveratrol or something
Right.
that's out there and uh, people are looking at as in clinical trials, there are like 20,000 papers, whereas psilocybin, there are not that many papers. I mean, maybe 300 studies ever in published in the United States. And but what I found was that, wow, this is an interesting drug. It is being used for so many different disease indications, everything from anxiety to depression, chronic pain, so many different disease indications. And it had been in nearly 150 clinical trials, and we still don't really know much about it. We know a lot about it, what it does in the brain, obviously, because it's known for its hallucinogenic impacts. And we know a lot about clinical outcomes from these trials that it has been in. But aside from that, there really isn't a whole lot known. And one of the things that I found intriguing was the so much information about the durable impacts, meaning like if you just take one dose,
Mhm.
people reporting positive impacts years later. And so I was like, how is that even possible, really? Because it's in and out of your system relatively quickly. We know a lot about the half-life of psilocybin, so I know we know how long it stays in your system. So how can it have these effects that last years? And so that was intriguing to me. I was very curious personally about how that could be possible. And so one idea that was out there was that, well, maybe it impacts aging. And I thought, well, I study aging. I can actually look into this.
So
It's kind
before we get
of
to the results, I've had several aging researchers on before, and I know that this is a very general question, but before we dive in, how would you personally define aging? How does the field think about it, like in terms of, what exactly is it? Do you have the sort of David Sinclair epigenetic lens on it? Do you have a different view on it? How do you personally think about aging at a high level?
Yeah, well, aging is a process. It's a normal, natural process that happens. And aging is not a disease. But at the same time, there's overwhelming evidence that aging contributes to worse outcomes in a number of different diseases, right? And so it's interesting to think about, like, we can target, one idea is, and a lot of my career is really targeting, like, okay, well, really looking at age dependent dysfunction. And that's really what my professional career has been about is, okay, well, this is broken in normal repair, this is happening normally, this is functioning properly. But in aging in disease, this is, let me figure out exactly what's broken and try to fix that one thing. But another way to think about this whole process is, what if you just slow down? What if you actually just target aging, and then instead of looking at just one individual disease, you can you can have better outcomes for a number of different diseases. So I started thinking about aging more as, a process that we can target and that could impact a number of diseases versus trying to figure out maybe what's wrong in one individual disease and trying to fix that one thing versus targeting aging as a process that you can impact so many more diseases potentially.
Yeah. So if aging is very much tied up with the body's natural reparative abilities, generally speaking, the longer you can keep those working well, the more buffered basically you are going to be against a whole wide range of diseases.
Right. Yeah, that's kind of my thought process now.
So let's talk about some of the so-called hallmarks of aging to get people thinking about some of the stuff that you measured in the study and just think about, what aging is at a cellular and molecular level. What is the relationship, generally speaking, between aging and oxidative stress and reactive oxygen species production?
Yeah. So oxidative stress is so oxidants in general. There's something that we need to function. We need oxidative signaling. It's something that's normal, natural, part of a lot of physiological processes. But it's one of those things like when you have too much, it's bad. It's if you don't have enough, it's bad, and if you have too much, it's bad. There's sort of like this optimal level of oxidant signaling that's good. Now oxidant, I think of oxidants as like rust to your body, and so over time this rust accumulates, and so oxidative stress just normally accumulates over time with age. So, and why is that? Different reasons. I mean, there are a lot of ways that can happen. I mean, part of it is that oxidants are produced from so many different angles, right? Like environmental stresses. So what you put into your body, the environmental stresses you have, aging itself causes, you have different things that aren't working as well that contribute to oxidant accumulation. So your antioxidant defenses don't work as well. You may produce more oxidants as we age, but the net effect is that you have this kind of accumulation of oxidative stress that happens over time. And so older individuals have more oxidative stress than younger individuals.
Mm-hmm. So basically, the level of reactive oxygen species production and oxidative stress tends to go up as we get older.
Yeah, and oxidative stress, oxidative stress is really that balance between your oxidant production and antioxidant responses.
Right.
So both, your antioxidant responses become reduced or less efficient with aging. So that alone, you have less ability to combat oxidants that are produced. And then you may have more oxidative damages or injuries or things that are contributing to more oxidant production. So overall, you have this kind of accumulation of oxidative stress that again increases with aging.
Mm-hmm. And some level of oxidative stress is inevitable because of how our metabolism works. We need to breathe oxygen, we need to burn it to create energy. And I guess just like, maybe this is an analogy, we often use fire in our homes. You might have a gas stove, you might have a fireplace. That fire is very energetic. You can use it to do work, but it's contained and controlled. And if it gets out of control, then you can start to do damage to the house.
Exactly. Yeah. And oxidants are highly reactive too. So, I mean, it's like the second they're generated, they are causing damage to surrounding molecules.
Mm-hmm. So another thing that people often talk about in the context of aging and how to measure it is the telomere length. So what are telomeres and what exactly is the relationship between the length of the telomere and aging?
Yeah. So telomeres are like the caps, the protective caps on the end of your DNA, and so the analogy I like to use is like those, uh, that little plastic piece that's on the end of your shoelace, that little cap. And when you buy that new pair of shoes, that cap is intact and long. And then as you wear those shoes, that cap tends to kind of fray at the ends, shorten. And
Yeah.
so that's exactly what happens to your telomeres, this cap. And so over time, every time your DNA is dividing, the ends of these caps start fraying a little bit more, a little bit more. So it's a natural hallmark, a classic hallmark of aging, because as we age, obviously our cells are dividing more, and that DNA is becoming compromised. These caps that protect them are becoming shorter. And so that's actually how we can measure, in some ways, biological aging by how long those telomeres are over, there is sort of an average telomere
Yeah.
length. As we age, we know it becomes shorter.
And so what exactly is the telomere? Is it a protein cap? Like what's it made out of?
uh, I should know this, but I don't know off the top of my head.
But it's sort of like if you ever bought, like I remember in my life purchasing cheap rope to go camping or something, and it doesn't have like the cap on your shoelaces, so it doesn't have a telomere, so to speak, and it just becomes it's frayed and unspooled until it's just useless as rope. And so the telomere is basically preventing that type of thing. Let's say.
Yeah, it's preventing this kind of it's protecting that DNA.
Mm-hmm.
It's protecting it as it's dividing.
Mm-hmm.
So it's think of it as like a protective cap.
Yeah.
And that protective cap is shortening. So
Right.
it's offering less and less protection over time.
Got it. And that just generally happens across species. The older the animal gets, the more the telomere is going to shorten over time.
Correct.
And then what about the SERT proteins? Where do they come into this? Those are famous in the aging literature. What exactly are those?
Well, I mean, David Sinclair's big on the SERT. I mean, these are sort of they do so many things. So I mean, I could probably spend a whole lecture telling you all of the different things, but they've been called kind of master regulators of aging. They have so many different functions, but we just we know from lots of research that's out there that these kind of master regulators are good hallmarks of aging and the aging process.
Yeah. And so set up the psilocybin study for us. What's the basic experimental setup for the in vitro work you did? You're looking at cellular aging. So you're looking at cells in a dish. What are the cells you're looking at? And how do you, just in general, how does one look at this type of thing in this field?
Yeah, so we didn't want to reinvent the wheel. We wanted to kind of use the tools that we have. And one of the tools that we have is these cellular models of aging. So there are different ways to study cellular aging. One of them is this cellular model of replicative senescence. One thing we know about cells is that they can only divide so many times. And so cells have a normal natural life cycle where they are dividing rapidly, and over time, they just, they divide a finite number of times until they stop dividing, they become senescent, and then eventually die. And so we can actually follow that cellular life cycle. And so the experiment was really simple. It was taking psilocin, which is the active metabolite of psilocybin, which is what is when you ingest psilocybin, it's broken down into psilocin, and that's really systemically what your cells in your body are seeing. And so we treated cells with vehicle or psilocin over the course of that life cycle. And it was, again, a simple experiment. It was like, if you give it, we're going to watch cells as they normally go through this life cycle, and we're going to treat them with psilocin or vehicle and just watch what happens to their cellular life cycle. And what we found was that, wow, we had a dramatic increase in cellular life extension, and that was dose dependent. So meaning the more psilocin we gave, the greater the life extension. And we did that in lung fibroblasts at first, and then we validated that in skin fibroblasts. So we showed in two different cell types that we were able to have this very similar dose-dependent effect.
I see. So the basic idea is you can culture cells in a dish, fibroblasts in this case, it could be other cell types. And in general, you put cells in a dish and you give them what they need to survive. They will divide some number of times, and you can count how many times each cell divides before it becomes senescent. You'll get a distribution of cell cycles, and then you can subject them to a different condition. You add a drug like psilocin or whatever, and you just see how that's affected. They might go from dividing, 20 times to 30 times or whatever it is.
Yeah, and we can count all that, how fast they're dividing, how many times they've divided, how many total cells are there. And essentially from all of that information, you can come up with your curve and see the population doubling curve, and then you can actually even tell, like, this is the point of replicative senescence. Like the cells have now stopped dividing, they're no longer dividing anymore. And so that where the curve just stops, the curve it becomes straight again. So so that's essentially what we're doing, just literally watching this treatment versus the normal cell life cycle and how and what happens. And again, yeah, we saw in two different cell types that we had this dose-dependent dramatic impact on cellular life extension.
So how many different doses did you use, and how big was this effect? Were the cells dividing twice as much, 10% more?
Yeah, so it's actually a really labor-intensive process
Hmm.
to do this curve. So for different things, we use different doses, but to quantify cellular life extension, we showed the data with two different doses. I can't remember. I have to go back and look at the paper, remember the specific doses, but I think 10 and 100 micromolar, we saw, anywhere from like 29% increase in life extension to like over 50% increase in life extension. But we tested different dose, a lot more of the range when we started breaking this down and looking for understanding the molecular mechanisms.
It sounds like you're seeing an effect here that's relatively large for this type of study.
Yeah, I mean, there's no question. Like we were like, wow, not only significant, but I mean, it's like one of those things where, yeah, sure, we can show significance with. It's always good to show significance with statistics, of course, but when you can see it visually with your eye, it's
Right.
obvious. It's like, yes, we of course back this with statistics, but the effect was very dramatic.
Yeah. And so psilocybin and psilocin, they're famous in the context of the nervous system for their psychedelic effects. People always talk about the serotonin 2A receptor as being sort of the psilocin receptor. But for nearly any drug like this, they're going to have lots of different things that they interact with in different cell types. Do these fibroblasts express serotonin receptors? Is that how psilocin is acting here? Did you figure out anything about what psilocin is doing at the cellular
Yeah,
level to result in
yeah.
this?
So yeah, good question. And actually, I knew nothing about I mean, I knew absolutely nothing about serotonin receptor or psilocybin for that matter when I started doing this work. So the first question I had when I started was, do these cells even express this receptor? Because you think serotonin, you think brain, right? I think
Mm-hmm.
most people would think that. And yeah, to my surprise, actually, most cells in the body express the serotonin receptor
Mm-hmm.
to different extents. So yes, fibroblasts do express the serotonin receptor, the same receptor that we know that it interacts with in the brain. Um, Many other cell types do as well. Now, that doesn't mean, and we're still working this out, is we don't know. Is it working on other receptors? We don't know. We're in the middle of those experiments.
Right. So these cells, so even though we're not talking about neurons here, these cells, and indeed many cell types beyond neurons, have expressed serotonin receptors of different kinds to different degrees.
Yes, absolutely.
And so what did these cells look like in terms of anything else that you looked at, any of the hallmarks of aging that you might have measured, reactive oxygen species production and those types of things?
Yeah, so we looked at a number of different markers of aging, right? And oxidative stress being one of them. I happen to be, work a lot on oxidative stress, so that was of interest to me. We saw that so the net effect as was reduced oxidative stress, and I mean when I say reduced oxidative stress, I mean oxidative stress levels that were actually significantly lower than even young cells' oxidative stress
Hmm.
levels. So it had a very dramatic impact on the amount of oxidative stress.
So,
And
so you basically had at the time that you would consider these cells to be old in the dish, in the presence of psilocin, oxidative stress levels were lower than what you would call young cells.
yes, yes.
Wow. And how do you measure oxidative stress? Like literally, what's the quantification there?
Yeah, so we just measure, reactive oxygen species in the culture media. So
Mm-hmm.
we can take this culture media and measure how much ROS, reactive oxygen species level, are there by a quantitative assay, and show, okay, this is how much oxidants the cells are producing.
Got it. So there's in vitro assays that are directly detecting ROS production.
Yes, that's exactly what we use, is like this, assays that directly measure the reactive oxygen species present.
Mm-hmm.
So that means it has to be produced by the cells. Now, we also looked at markers of oxidants, right? So we found that NOx4, which is a master regulator of oxidant production in many cell types, that's its only known function is to produce oxidants. NOx4 levels were very, very low in these psilocin-treated cells. And NRF2, which is a master regulator of hundreds of antioxidant gene responses. NRF2 levels were extremely high. So you had, the net effect is you have much greater antioxidant responses and much lower oxidant production. And that net effect is reduced oxidative stress.
Got it. So markers change such that in the presence of psilocin, there were fewer oxidants and more antioxidant production. So basically, the cell can tolerate more oxidative. It can tolerate more oxidative stress, or it can protect itself from that stress for longer.
Right. Exactly. Yeah.
Interesting. So what about like telomere length or anything like that?
Yeah. So telomere length was preserved. So whereas, again, we normally see that classic hallmark of aging that you see telomere attrition. So telomere length is shortening over time. So in cells that received vehicle treatment, we compare these back to the same cells as young cells. We can see that telomere length was much shorter with normal aging, and that shortening of the telomeres did not seem to happen to the same extent in psilocin-treated cells. So there was so that telomere length was preserved when you treat with psilocin.
So you looked at a number of things: reactive oxygen species production, the expression of regulators of oxidants and antioxidants, telomere length, a lot of the major things one would look for. Yeah. And so basically across the board, psilocin made these cells less senescent by increasing antioxidant stuff and decreasing oxidant stuff.
Sure, yeah. And then, we see these other, and so now the question is like, how are these things working? Because, we don't think, for example, we have some unpublished data showing it's not acting directly on NOx4, but the downstream impact is on NOx4. So psilocin doesn't, we don't think it's binding to NOx4 and doing something, but the downstream effect is that it's impacting these pathways, right? So we don't know the order of what's happening first.
Right.
How these things are interplaying.
The end effect is this anti-aging effect. You know that psilocin is getting in there somehow, but it's a bit of a black box in between.
Right. Like for example, is it the fact that you have less oxidative stress? Is that why the telomeres length is preserved? Or is it the fact that you have more, or is it the other way around? Do you have more protection of your telomeres?
Right.
And that's leading to more efficient DNA repair, and that's what's causing the net effect of less oxidative stress. How those things play into each other, we're still kind of teasing out. So we don't.
Yeah.
But the net effect is that you have that the cells will age with psilocin treatment, but just slower. So you still have the cells will still become senescent, and they will reach the peak of their replicative senescent lifespan, but just that process is delayed when you treat with psilocin.
Right. And so a natural question at this point is, if it's having this effect in a dish, would it have this effect in a whole animal? And so what did you do there?
Right. So yeah, we were completely excited about these findings in cells, and so of course the next step is, well, we need to test this in animals. And so when we were thinking about this, we had limited pilot funding that was generously provided by Emory University, where we initiated these studies. And we had to think carefully about, given kind of, the feasibility, what we could do with a limited amount of pilot funding to actually do this experiment.
Yeah, because now you got to house the mice, and it's an aging study, so you got to keep them around probably for a long time. And for those who don't know, it gets pretty expensive pretty quickly.
Yeah, it's a long study because this was a year-long study, but also people probably aren't. At least, I don't know, people who don't do aging research might not realize how expensive aged mice are. They're like, $500 or $600 a mouse, right?
Yeah. And like for for a normal, I should say, a non like longevity study, you might use a mouse that's a few months old, or you might have the mouse for a few months of its life and then you're not using it anymore. But for, as you're going to tell us, right, you're keeping these mice around for a year or longer.
Yeah, so we started with aged mice, so they're expensive to procure in the first place, right? And then we had about 30 mice per group, and then the reason we use aged mice because we wanted to have a clinically relevant testing protocol. We wanted something that could, be a starting point for translation in human patients. So what does that look like? Well, let's try a therapeutic intervention when we have mice that are the equivalent of human age 60 to 65 years old. And so that's what we did. We designed an experiment where we gave the mice high dose psilocybin once a month. And we did that for a total of 10 months.
And remind me, how old are the mice at the time the study starts?
Yeah, they were 19 months old.
And can you give us a sense for like, what is that? Is that an elderly mouse?
Yeah, that's about like 60 to 65 years, human years.
Got it.
Yeah. So we started with, these older mice and once a month treatment, and we monitored them very, very closely over the course of that 10 months. And now I know you mentioned the word longevity. So this wasn't a longevity study. We weren't actually allowed to do a longevity study per the guidelines of our animal care protocol.
So you couldn't allow them to live as long as possible.
Right. And that's because at the time, there were no research ever conducted on psilocybin and aging. All of this study was being done based on our unpublished data in cells. And so, there are concerns from animal care about the Schedule 1 drug, giving it high dose for the rest of the animal's life.
Yeah.
Are there potential harms?
Yeah. For those listening who don't know this about animal research, you can't just do whatever you want to animals. There are standards and limitations and a lot of regulation there.
A lot of people weigh in on our study design, and for anyone that does animal studies, these are important considerations in thinking about the welfare of the animals and what is a reasonable study based on the information that's out there. And so we decided that the most reasonable study was to start with aged mice and then give it for as long as we could. The sort of the end point for us was when any given group reached 50% mortality, that signaled the end of our study.
Got it. So you're giving retirement age mice, let's say, a relatively high dose of psilocybin. Before we get to the result, you're giving it once a month. And so what is a high dose here, and how are you administering it?
Yeah, so a high dose was 15 mg per kg in the mice. I know a lot of people are kind of constantly asking me this question about like, well, what does that mean? And how did you decide this dose? And how does that translate? And, it's complicated because we took a lot of time to decide, how to set up this treatment protocol.
Mm-hmm.
And in part was based on a lot of different, many factors, but in part on what's being done in clinical trials with high dose.
Yeah.
Kind of looking at high doses of being administered to patients that as kind of a starting point for thinking about dosing. But another consideration that we had to factor in is that we do know a lot about the pharmacokinetics in mice versus humans and the metabolism of mice versus humans. So mice have much higher metabolism. They can eliminate psilocybin like 4 times faster than humans. So we took all of these factors into consideration and came up with this dose. So again, I know people are always asking like, well, what does this dose translate?
Right.
It's not that simple. There's a lot of factors that you need to think about when directly translating.
Yeah. For those listening, like what we're getting at here is I've done a podcast with people like Jeffrey West before, and metabolism doesn't scale linearly with body size. So it's not just straightforward, like this animal's 10 times smaller, so you get 1/10 the dose. It's a little bit more complicated than that. And basically mice have a much higher metabolism than we do. And so you have to take that into careful consideration when thinking about the dose. But 15 mg per kg, this would be roughly, this would correspond to a hallucinogenic dose. I don't know if that would be a heroic dose in a human, if you did the math, but it would be a psychedelic dose.
Absolutely. Yeah. Yeah. And that's what we were going for, because I think most, at least from what I know, most of the clinical trial data that's out there, for the most part has, or at least more than microdosing, we know more about the high dose
Mhm.
psychedelic dose.
Mhm.
There have been more clinical studies with the high dose. So I wanted to start with that, just because from a clinical standpoint, we know more about that.
Yeah. Oh, sorry. Final question, and this is an important one before we get to the result here. What would be the average number of months that such a mouse would be expected to die?
Yeah, so mice tend to have their median survival is around 30 months.
Mhm.
And so, anywhere in that vicinity. And so we expected that this would be approximately a year-long study.
Because
So
you're starting at 19, I think you said, and then you're going,
yeah,
you know that you're going to go roughly that long.
right, right, yeah.
Okay, so psilocybin versus vehicle. What did you see?
Yeah, and so we treat by oral gavage, which is basically orally treating the mice. And yeah, so we initially we saw that, again, because we've tagged these mice, or have tags. We are literally monitoring each individual mouse on an almost daily basis, and we started seeing it right away, very early on. We started seeing that the mice just seemed to look better. They seemed healthier, and I don't know exactly how to quantify that, except for that it's something that all of us in the lab seemed to notice right away.
Yeah,
Uh,
you have a picture in the paper, and it doesn't take a trained expert to see that there's a difference here. Like,
yeah,
the fur is shinier. Everyone
yeah,
knows what like an old dog and a young dog look like in terms of just their
right.
general anatomy and structure, their gait, their fur healthiness. Like, yeah, these mice definitely look different.
Yeah, and it's some of these things, honestly, again, I don't study mouse fur, skin, hair quality, things like that. So some of these things I really didn't at the time know how to quantify. Things like you said, like shiny hair, and you can see the old mice have much kind of scruffier looking hair. So there were a lot of things that scientifically I don't have data on, because I just literally did not know how to quantify.
Right, right.
That doesn't mean that I didn't see these things and that
Yeah, yeah,
whole lab didn't also observe these things, like shinier hair and sort of less scruffiness of the hair. But the things that we were at least able to show visually, and the best I could do at the time was photographic evidence. And
right, right.
you can see, hey, well, look, this mouse that had a bald spot has grown back its hair, and this mouse that had white hair
Yeah.
has, and
Yeah,
that
so
white hair.
it's not just like they didn't look as haggard in the end. You started with old mice, and in the
Right.
psilocybin group, some of them already had patchy and mangy fur,
Yes.
and it actually improves over the course of like 10 months.
That's the craziest part, is not only did they look better than the vehicle mice, but they looked better than they looked at the start of the experiment. So
Right,
they looked
right.
younger than when they started at the end of this experiment 10 months later.
And so what's the headline result here in terms of this survival curve?
Yeah, so when 50% of the control mice had died, only 80% of the psilocybin mice were still alive. So there was a significant improvement in survival.
Mm-hmm. So you're looking at the mice from about 19 months, which is already old. That's a 60 to 65-year-old human, roughly speaking. By the time 50% of the normal vehicle treated mice die, you're at 28 months. So half of them are dead. At that point, almost all of the psilocybin mice, 8 out of 10 of them, are still alive.
Correct. Yeah,
80%, there's more than 10.
yeah,
Wow. Yeah.
80% still alive versus 50%.
Yeah. Wow. So that would be, and in this field, that would be a pretty big effect size, I would imagine.
That's a pretty big effect size, because I will say too, and anyone who does aging mouse studies knows that starting with 30 mice per group is a very low number. Typically, you would need 100 mice per group to
Because the
power.
effect will be smaller and you just need more to see it.
Right,
Got
right.
it.
And just in general, anyone who does aging research knows that there are a lot more variability. Like, if I do, even with young cells or old cells, you see like the error bars are always tight with young. anything, cells, animals, in anything aging related experimentally, just the error bars are higher. There's just more variability when it comes to aging studies.
Mhm. And were you able to look under the hood at all in the in vivo experiments and see what the cells looked like or anything like that?
Yeah, so we didn't publish on this, but we collected so much data from this, and this is something that we're still kind of sifting through. So we've collected, and we took like an omics approach, and we have collected everything that we could collect. And so, um, it's actually so much data, and I'm not an omics
Right.
person, or at least in class, my training isn't in
Yeah.
that.
So
So
you're probably working with other people and that
yeah.
will come at some point, but you collected tissue samples. You're probably looking at gene expression across the whole
Like
body and things like that.
everything, yeah. So we can tell you what's happening in every organ across the body and what pathways are up and down. That's what we're actively doing that now. So we'll hope to have more for you soon, but it's complicated because it's big data,
Right, right. Yeah, so it's going to take time, but I mean, you must see effects across many different tissues. I mean, if you can see bald mice growing their fur back, there must be something going on in the hair and the skin. It's not just going to be the brain. It's probably
Oh
going
yeah,
to be lots and lots of different changes all over.
absolutely, yeah. Yeah.
And so, well, I was going to ask you what comes next, but it sounds like that's probably what comes next.
Yeah, I think there's so much. I mean, of course, any scientist, like anytime you answer a question, you just end up with 100 more questions. And so we have a million more questions really after this study, because the findings were so incredible, that I just want to know more. Everything from how is it working mechanistically, and we're doing sort of the classic studies of blocking receptors and looking to see if we still get you know uh, the same impacts on in cells, cellular life extension,
Mhm.
to how do you define the optimal treatment protocol? I mean, what if you started giving it at
In
two
earlier age.
months of age in the mice? Like, do you see tenfold greater survival? I mean, we just don't know, right?
Right, right,
What if you give it what if you gave it to mice that were equivalent of 80? Do you see any? Is there a point
right, right.
in which you don't see any impact? Is it too late at some point? We don't know that. What about, we're also, some of our unpublished data indicate that the frequency of dosing is really important actually
Got
for
it.
this.
And
So
remind me, you said you were giving these old mice a once per month dose. So you're talking about roughly 10 doses in the psilocybin group.
yes. Yeah.
Wow. Yeah. So it's not every day. Because when you start to think about like, will this translate? What would that look like in a human being? They're not tripping every day. It's a once per month thing starting at this pretty advanced age.
Right. So that could be more along lines of once a year in humans.
Right, right, right.
We're
Interesting.
noticing that frequency of dosing has some interesting impacts, that it may do different things at different doses, low and high doses, and even the frequency of which you dose may have different impacts as well.
Yeah. And so stepping back, when we think about aging and oxidative stress more generally, and we think about fibrosis in particular, and just, the scarring that tends to happen in tissues and organs as we get older, I'll let you drive here in terms of, what you focus on, but like, what are some of the organs where you see the highest levels of fibrosis in humans? What organs is it the most problematic in? Is it the heart?
Yeah, that's a good question. Yeah, I mean, yeah, that's a really good question. I think there's a difference between, like, so organs in general, you're going to get, there's increased just kind of fibrosis in different organs as we age.
Mhm.
But then you have like fibrotic disease, um, that happens. Like, for example, I study idiopathic pulmonary fibrosis, which again is a rare disease. It is the most progressive and fatal form of scar tissue, which happens in your lungs. We don't, by definition of its name, idiopathic, we don't fully understand, how you get
Yeah, yeah.
disease.
But someone with this disease has just way too much scarring or way much
Yeah.
faster scarring than other people.
They basically, the average age of diagnosis is 66. So it's a disease of the elderly population predominantly. And then these people that are diagnosed with this idiopathic pulmonary fibrosis, or IPF, um, they have this rapid and progressive scarring of their lungs. And so there are FDA approved drugs that can slow the progression but can't stop or reverse the progression. Um, and so yeah, the holy grail of this, research to me is you know, can you give this as a therapeutic agent that could reduce these age-associated disease outcomes?
Mm-hmm, mm-hmm.
So like,
Right,
so
because
maybe,
you not only saw the aging effect in mice, but you saw the cellular senescence effect in fibroblasts, and so
right,
it's possible that it's going to affect exactly the cells that are responsible for the scarring.
right. So yes. And so maybe you're giving these cells the ability to again have additional plasticity and be able to repair better. We don't know the answer to that. That's one of the things that I'm really excited about. Again, the potential, you know, applications of these findings, to me, that would be the most exciting is, can we use this to improve age-associated disease outcomes?
Yeah, yeah.
That'd be, I mean, wow, if we could do that, that'd be incredible.
And when we think about aging generally, but also what the fibroblasts are doing and scarring and fibrosis, I would imagine that, because I think one of the best predictors of how long someone lives is actually how long their parents and grandparents lived. Is there a lot of natural variation in the sort of scarring side of this and what the fibroblasts are doing that just varies a lot between people?
Yeah, I mean, it's a good question. I mean, sure, patient to patient, of course, there's so many factors, right? And we know that there are genetic risk factors. So
Mm-hmm.
there are absolutely genetic risk factors, but then there's also external risk factors like smoking or, being a being being in like, we don't think of these things as injury, but like, let's say you were present during like a tornado or hurricane,
Mm-hmm.
that can actually cause a lot of lung injury. Asbestos, so there are a lot of ways to injure the lung, or these injuries that could have even happened a long time ago that are
Yeah, yeah.
contributing. So there's so many factors, which is why it's tough to study the differences in
Right.
patients, right? But
Right.
yeah,
But at a high level, it sounds like if we're talking about the lungs, say, every time that you breathe in irritating stuff that can damage cells in the lung, that will be repaired to some extent. And every time that happens, you have those fibroblasts that are helping repair the damage. They're dividing. And with every division, that's one step closer to senescence. And the
yes,
more that happens, eventually you're going to get more scarring, over scarring, and then,
sure.
it becomes a visible problem.
Right. Yeah. So, I mean, again, or scar tissue is an evolutionarily conserved process. We need
Right.
it to survive. If you could not scar, you wouldn't be here today.
Mm-hmm.
So we need it. We need that process. But when it becomes a problem is when you have a persistent and continued accumulation of that scar tissue.
Mm-hmm.
And so that's when it becomes pathological. So it's a physiological process until it be in, and then it can be also a pathological process.
Yeah, yeah. So it's like oxidative stress in a way where
Exactly.
it becomes problematic at some point, but we're talking about a very fundamental thing that you need for survival as an animal.
Right.
Are there any final thoughts you want to leave people with, or anything you want to reiterate from our discussion, whether it's about the psilocin study specifically or about fibrosis and aging and oxidative stress generally?
Well, I think these are really exciting findings. I hope that it opens the door for so many other people to come in and think about psychedelics in a different way. I mean, we've always really classically thought about psychedelics, about what it does in your brain, and I think this really opens up a new frontier and thinking about these things differently, that so many other cells express these receptors that they work on, and to think about the potential of psilocybin in different ways that we've never really thought about. People are say they commonly use this term like plant medicine. It's a plant medicine, but it's actually not a plant medicine because in there's five like classifications, plants, animals, protists, monera, and fungi. Fungi is its own kingdom. So it's like this whole other entity that we just don't know enough about, I think. So I think it's exciting, and I hope people start thinking about it in a different way that we haven't thought about it before.
All right. Well, thank you for sharing all this. This is fascinating and exciting stuff. And it sounds like we've got a lot more data coming that's going to be, very quantitative and wide reaching in terms of its scope. You looked at a lot of different stuff, it sounds like. So it'll be really interesting to see that when it comes out. Louise Hecker, thank you very much for your time.
Thanks for having me on your show.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about one of my cool new affiliate partners called Seed Oil Scout. Seed Oil Scout has a really cool map feature that helps you find restaurants in your area where you can find seed oil-free options. They also have a really cool product scanner, so you can take out the app and use the camera functionality of your phone to scan products in grocery stores and see what's actually in these things. Do they contain seed oils? Do they contain other things that you want to know about that aren't necessarily on the box? This will help you find places to eat and find foods to eat that actually have what you want in them and help you avoid those that don't. So if you're looking to cut back on your seed oil consumption, or you just want to know more about what's in the foods on the shelves at the grocery stores, download Seed Oil Scout today. If you go through the link in the episode description, click into that and you'll see a link to the Seed Oil Scout app. If you go through that link on my support page, you'll be directly helping the podcast, and you'll be able to download this really cool, easy to use app that will help you find healthier food options. So go through the link in the episode description and download Seed Oil Scout today.